Hello, everyone. This is our research presentation on genome variation analysis between different American ethnic groups to improve precision medicine. So first, I'll go a little bit into the background of our research. So our research looks at the field of precision medicine, which is a rapidly emerging
Risha,
research field.
Risha, please speak more slowly. You're going 100 miles an hour.
So our research project specifically looks at precision medicine, which is a rapidly emerging research field that focuses on personalizing or catering medicine for people based on their specific genomic makeup or genotypes. Genotypes are basically the specific set of genes or variation of genes, known as alleles, that influence the traits in individuals, such as eye color or if they have dimples or not. So what we're looking at specifically is how do various genetic mutations or variations in the genes, as well as environmental factors for humans, affect their susceptibility to diseases, as well as how well they metabolize certain drugs. So when conducting a literature review, we noticed that certain studies have shown the need for precision medicine because there are various different ethnic groups that have different levels of these genes or different levels of these alleles, so the same particular medicine can't be used for each of these ethnic groups. For example, we found that different ethnic groups in northern Thailand have varying frequencies of the allele for alpha thalassemia, and alpha thalassemia is a blood disorder common in Southeast Asian people. So there is a different varying level of these alleles within these ethnic groups. And then we also found that indigenous Australians as well have a lot of genetic variation or a lot of different alleles in their populations as well. But there's more research needed on this because indigenous populations are quite underrepresented within the field of precision medicine. So a research question that we're trying to answer is a personalized medical approach required amongst different American ethnic groups due to different genetic variation? So the purpose of our study is that a lot of the time when we're conducting our literature review, there's a huge lack of diversity in studies done on individuals other than those of European ancestry. So in our study, the novelty is that we're using different individuals or indigenous people because they're very underrepresented within the field of healthcare. So our data comes from particular indigenous populations. We are using data from the European Nucleotide Archive, which are 47 samples from Harvard Medical School. And the Paleo-Eskimo people are indigenous peoples from Northeast Siberia. Our data is publicly available and found online, and it's also about 1,000 to 5,000 years old. But we found that it's genetically linked to modern day indigenous individuals like the Inuit and Yupik peoples.
So now we'll be giving an overview of our methodology. So first, we conducted epidemiological research to identify health disparities and determine where precision medicine would have the greatest impact, which includes studying the prevalence of diseases. Next, we used variant calling to identify mutations within the genomes of modern Arctic populations. This was followed by variant annotation, which classifies these mutations and helps us understand their biological implications. This was followed by pharmacogenetic analysis, which links genetic mutations with clinical implications. And finally, we found genotype frequencies, which is just finding how common each genetic variation is within a population. So for reference, epidemiology is the study of the prevalence and causes of diseases and health conditions in populations. For the study, we analyzed 47 ancient Paleo-Eskimo samples originating from regions like Alaska, East Siberia, Chukotka, the Aleutian Islands, and the Canadian Arctic. These samples are genetically linked to modern-day indigenous populations of the Arctic, such as the Yupik and the Inuit. By connecting ancient genomic data with modern epidemiological research, we aim to uncover health risks faced by Arctic indigenous populations. This enables us to apply significant findings and make clinically relevant recommendations that can improve healthcare outcomes in these communities. So across Alaska, East Siberia, and the Canadian Arctic, the most prevalent health issues include cardiovascular diseases, obesity, diabetes, respiratory illnesses, TB, and mental health disorders. These conditions are statistically more prevalent among indigenous populations than their respective national averages, often due to environmental, genetic, and societal factors.
So after our epidemiological research, we perform variant calling on our samples, and variant calling is just the process of finding the genetic mutations or the variants in our data. And this first part of the pipeline shown on this slide was performed individually on each of the 47 Paleo-Eskimo samples. So our pipeline was based on the GATK best practices for variant calling and the 1000 Genomes Project Phase 3 SNV and indel discovery pipeline. And so what we do is for each sample, we have a FASTQ file, which is basically like a text file of the short raw DNA sequences that have been read by a sequencing machine. And we align these sequences in the FASTQ file against our reference genome. And a reference genome is like a standardized genome that is used as a point of comparison. And so for alignment, we use a tool BWA-MEM, and it'll just take each of those individual short sequences, and we're not sure where in the genome they're from, and it'll map it against the reference genome so we have a complete picture of how our data looks as like a full genome. And this outputs a binary alignment map or BAM file. And then we run a few more processing steps on this BAM file. So we sort it, we add read groups, we remove duplicates, and we also run base quality score calibration, which is a machine learning algorithm which adjusts the quality scores. Quality scores are just outputted by the sequencing machine, and they represent how confident the machine is that it correctly read a DNA nucleotide. And so these quality scores will later on affect how confident we are in our variant calls. So after these initial processing steps, we have a binary alignment map or BAM file that's ready for the actual variant calling. And we use GATK haplotype caller in GVCF mode for variant calling. And so what this does is this outputs a GVCF or a genomic variant calling format file, which contains at every position in the genome, both the variant positions and non-variant positions, and it has likelihoods of genotypes for every position. So at this point, when we're doing this process individually for every sample, we just have the probabilities for genotypes and not the final called genotypes. So in the next step of our methods, we run joint genotyping, which is analysis of all our 47 samples together. And this is where we call the final genotypes for every sample for every position in the genome. So we input 47 GVCFs, which were the outputs of our previous step. And as we run them through our joint genotyping pipeline, which is also based on GATK's best practices, we, in the end, have a file of variants that has the variant data for all samples across all chromosomes. So we have all our genotypes, and these will be used in the future for our analysis.
So this methodology focuses on finding genotype frequencies, which is mainly used once we've identified the clinically significant or the variants that are highly prevalent within our population. So finding genotype frequencies is a method used to identify how common specific variants are within different populations. So for example, a single variant can have different frequencies and health implications across different ethnic groups. Genotype frequency data is also used in understanding the correlation between drug responses and a person's genetic makeup. So in this process or in this pipeline, there are two inputs, one of them being the file with the chromosome number and positions of the clinical variants, as well as the index file of the variants present in 26 plus samples. So our code starts off with extracting variants from the positions file and then using BCF tools to extract matching variants from the index VCF, which results in the file containing the extracted variants. This file is then used as an input for our finding genotype frequencies pipeline, which uses BCF tools to extract the genotype data and then uses a language called awk to process each variant line. So awk basically allows the user to filter, manipulate, or summarize data easily. And in our case, we manipulated the code so then awk can output the chromosome number, the position, the reference and alternate alleles, the homozygous reference, the homozygous alternate, and the heterozygous frequencies, as well as the miss rate. Once these variant lines are processed, then the code counts the genotypes for each of the variant lines and then calculates the frequencies, which outputs the file of genotype frequencies.
So now I'll go over our results. So among our 47 Paleo-Eskimo samples, we found almost 4.2 million variants. And this histogram shows the distribution of variants across samples. So each bucket in the histogram represents a number of variants ranging from 1 through 40, or sorry, number of samples ranging from 1 through 40. And the height of each bar or the y-axis represents the number of variants that are common to that number of samples. And so we should note that the graph is in a log scale, so the descent is actually much steeper than it appears. And we see that like most of our variants are just present in a few samples, But as we increase the number of samples, we find fewer and fewer variants are actually common to this number of samples. And actually, we found that none of our variants were present in more than 40 samples. And this is an expected trend because in general, genetic variants are fairly rare and not found like to be very prevalent in the population. But in our further analysis, we hope to focus on variants that are highly prevalent in our samples because they're then more likely to be present in the actual population. And so we had 4.2 million variants, but we want to understand how they actually affect functions within the cell. So we annotated the variants using the ensemble variant effect predictor or ensemble VEP. And VEP gave us information about the genomic location of each of the variants, as well as its functional consequence, which means how it affects transcription, splicing, translation, other related processes, and then the final protein product. So here we have the frequency of variants, or like how many variants are present in different functional consequence categories given by VEP. And so these categories are not mutually exclusive, so a variant can be present in more than one category. And this graph is for SNPs, or which are SNPs, or like single nucleotide polymorphisms, which are a change in a single nucleotide base pair. So they can affect the amino acid sequence, they could affect splicing, or they may have no effect. And we also analyzed insertions and deletions, or indels, which are just a nucleotide that has been either inserted into the DNA sequence or removed from the DNA sequence. And this could have no effect, or it could like totally shift the reading frame for when it comes to transcription and translation. So we also have the different categories for indels here. And this just gives us more information about what variants are like highly impactful when it comes to protein function versus some that don't have impacts.
We focused on variants that were highly prevalent in our samples, and we were looking for variants linked to drug therapy responsiveness. So from there, we narrowed the list of variants by reviewing ClinPGX annotations to assess their clinical relevance and compared these with our genotype frequencies. We also prioritized variants predicted to have deleterious effects using tools like polyphen and SIFT, since these are more likely to affect protein function and drug response. While there's no currently standardized statistical method to rank pharmacogenetic variants, our approach involves a comprehensive literature review based on drug metabolism and toxicity. So essentially, we cross-referenced each variant with the trusted pharmacogenetic guidelines like ClinPGX and the NCBI SNP database, in addition to pharmacogenetic literature to ensure that our findings are actionable. So this is a list of some variants within our population that are prevalent and are high at risk for adverse drug reactions. So adverse drug reactions are unintended and potentially harmful effects that occur as a result of taking a medication and can range from minor to fatal side effects. So we looked at specific variants, their effects on the drug based on the genotype and the prevalence of that genotype in our samples. So for example, if we look at RS316019 in gene SLC22A2, for reference, this gene encodes a protein crucial in the transport and elimination of certain drugs, especially chemotherapy drugs. So 75% of our samples that had genotypes called had the CC genotype. This genotype is linked to increased risk of cardiotoxicity with anthracyclines, which is a group of cancer drugs, and ototoxicity with cisplatin, a really common cancer drug. And also the CC genotype is associated with increased metformin clearance, which is a diabetes drug.
During this project, we've observed clinically impactful variants among our 47 samples based on the clinically significant variants and genotypes identified. We aspire for our findings to establish better health outcomes for modern Indigenous populations in the Canadian Arctic, Alaska, the Aleutian Islands, Chukotka, and East Siberia. Some limitations in our data include the sample size and sample age, and a problem that we faced during this entire process was finding modern day data about the Indigenous populations due to data exploitation, which explains why we focused on ancient data for our findings. So currently we are working on selecting the most significant variants to be able to run our different methodologies and to get more results that can help boost our project progress. And then we hope to analyze the effects of variants at a more molecular level. We would like to perform protein modeling on select variants with deleterious effects and compare select variants with global populations through statistical measures. We'd also like to replicate our methods using modern-day data from biobanks to derive more clinically relevant insights. We'd like to thank Mr. Kanha and ASDRP for the resources, support, and the guidance that they've given. Next are our references, and the floor is open for questions now.